Hello and welcome to our webinar. My name is Arturo Ramirez and I'm a senior scientist at RareCyte. Today I'll be discussing our work on analysis of circulating tumor cells in patient-derived xenograft models of breast cancer. CTCs, or circulating tumor cells, are cancer cells that are found in circulation that can originate in the primary tumor or in metastatic sites. Getting access to CTCs is minimally invasive. It allows repeated sampling and provides a real-time snapshot of how the tumor evolves. Because CTCs are whole cells, they can be assayed for protein, DNA, or RNA. Patient-derived xenograft models, or PDX, are tumor lines that originate in humans but are transplanted into mice and can act as surrogates for the original tumor. In this sense, they can be used to study the original tumor and its response to different treatments without posing any danger to the patient. RareCyte has developed a rare cell analysis platform that recovers over 90% of cells spiked in the blood. All nucleated cells in the sample are attached to glass slides where they could be automated staining and then scanned on our fluorescent instrument. Our instrument can image up to 6 markers simultaneously. 4 channels are used for the CTC identification, and this leaves 2 open channels for any biomarker of interest. Our instrument can also pick individual cells from the glass slides and deposit them into PCR tubes for downstream analysis. This slide shows the workflow for CTC analysis that's been modified for mouse blood. The first step is to do a red blood cell lysis of the small volume of mouse blood that's collected. Then, the cells are spread onto a monolayer on a regular microscope glass slide. From here, the slide can be stained on an automated stainer for different fluorescent markers. In panel D, I show our fluorescent instrument. It's the site finder. This instrument can scan up to 6 different channels and uses 4 of these channels to identify the CTCs of interest using algorithms that look at morphological and other characteristics of the cells to identify the cells. Once the cells have been identified, their precise location is known and can be used to pick the cells off of the slide and into individual PCR tubes using a CTC isolation device called the site picker that is inside the instrument. Before we started testing real PDX mouse models, we tested spike-ins of cultured breast cancer BT474 cells into 125 microliters of mouse blood. Here we found our recovery rate was about 90%. And as you can see, the cells can be identified by their DAPI staining, showing that they have a nucleus, human pancytokeratin staining that identifies them as epithelial cells of human origin, and they're negative for the mouse CD45 isoform. As a 4th channel, a confirmatory channel, we pooled EpCAM, HER2, and EGFR, although some CTCs were negative for this channel and were just identified with a pancytokeratin marker. The work that I'm going to talk to you today about was done in collaboration with Rachel Schiff, Meghana Trivedi, Michael Lewis, and others at Baylor College of Medicine who have published on 18 breast cancer PDX models and the presence of CTCs and how they relate to metastasis to the lungs. For this work, we tested a subset of these lines to enumerate CTCs and CTC clusters. We performed molecular analysis of single CTCs and studied the effect of chemotherapy on CTC number. The first PDX model I want to talk to you today about is the BCM4888 model. It was established from a tumor with a known mutation in PIK3CA. This tumor was found to be ER positive, PR positive, and HER2 positive. And in the PDX model, 67% of the mice had lung metastasis. We analyzed mice from the 16th generation of this model, and we analyzed 3 different types of samples: CTCs from circulation, primary tumor cells that were dissociated and spread onto slides, and lung nodules that were also dissociated and spread onto slides. Single cells were picked, whole genome amplified, and then PCR and Sanger sequencing for PIK3CA was performed. Here are the results of the Sanger sequencing. We can see that the known mutation that was found in the original tumor was also found in all of the CTCs, the lung nodule cells, and the primary tumor cells from the mice. Furthermore, the sequence that was found for PIK3CA corresponded to the human isoform and not the mouse one. In this next experiment that I'm showing, we tested two different PDX models, BCM4272 and BCM3887, which are both triple-negative breast cancer models. We treated these mice with either a vehicle, docetaxel, carboplatin, or the combination of both of these once the tumors had gotten to a certain size. As can be seen in this image, in BCM4272, the CTC number and the number of CTC clusters declined with docetaxel and the combination treatment, but not with carboplatin. In contrast, in the BCM3887 model, CTC numbers increased with docetaxel and the combination treatment. Further study needs to be performed to understand the implications of changing CTC numbers with treatment. Finally, I wanted to show you that our platform commonly identifies CTC clusters in circulation. From the literature, it's known that CTC clusters have a much higher metastatic potential than single CTCs. Here in this slide, I'm showing a CTC cluster of approximately 100 CTCs that was found in the PDX model BCM3887. Finally, I'd like to thank you for your attention, and if you have any questions or would like more information about our technology, please contact us at info@rarecyte.com. Thank you for your attention.